Welcome to the Year 10 marking video of the exam on controlling blood glucose. The first question asks you to name the organ that produces insulin, and the answer is pancreas. Remember, insulin is the hormone that helps to control your blood glucose levels. Now, it's very important not to get the roles of the liver and pancreas mixed up. So here's a quick guidance and reminder on what the roles of the two organs are. The pancreas produces enzymes and it produces hormones. The liver does not do either of these things. What the liver does do is it produces bile, which is a substance that helps digest fats. The liver also stores glycogen, which is a polymer of glucose that is insoluble. And the third thing the liver does is it breaks down waste products by detoxifying the blood. For example, if there are too many amino acids in the blood, they will be broken down by the liver into urea, which we then excrete as urine. So, the liver, although it produces bile, does not produce any hormones or any enzymes. But the pancreas does. It produces enzymes for digestion, lipase, protease, and carbohydrates. And the pancreas also produces insulin and glucagon, which help control blood glucose concentration levels. Let's move on to question 1b, which is about treatment of insulin. Although we haven't studied different types of insulin in class, that's not expected. What you are expected to be able to do in this question is to read the table carefully enough and be able to apply what you have interpreted from the table in exam questions. The first thing you should always do when you get a table or a graph is to cat the table or cat the graph. So you read the caption. In this case, it says the table gives you the properties of 4 different types of insulin. So I already know there are 4 types, A, B, C, and D, and I know that there's going to be something about the different properties of those insulin types. Next, you need to look at the axes. If it was a graph, obviously we don't call we don't have an axis in a table, but what we refer to is the table headings in this case. So we've got type of insulin, we've got the time taken to begin for it to begin to work in minutes, etc., etc. Read through that carefully before you start answering questions. Finally, look for trends. So I'm already noticing that D takes the longest to begin, it takes the longest to reach a maximum concentration, and it lasts the longest because it seems to be the one that takes up to 20 hours before it is no longer effective. So I already have a picture in my mind of what the different types of insulin do before I'm reading my question. So this one is asking you which insulin type is needed just before a meal to stop a big increase. So if it's taken just before a meal, it needs to work quite quickly if we want to stop a big increase. So which one works quickly? I'm going to look at my table. I think the relevant column here is the one that doesn't take very long to start, and it seems to be A. It only takes 15 to 20 minutes. So I'm going to pick A, and my reason is it works the quickest. Now, importantly, you must use comparative language in this because you are asked to choose from a list of options. Any questions that ask you to do this, you must use comparative language. Shortest, not short. Quicker, not quick. The second question asks you to find the type of insulin that will help keep the blood level, sugar level under control for the rest of the 24 hours. So it needs to be long lasting. Now I know that that's D by looking at the table because the column that is relevant is this one here. The time when the insulin is no longer effective in hours. Well, this one takes 20 hours before it's no longer effective. Therefore, it is D that is the correct answer. It acts for the longest time. Again, I've used comparative language. Now, the final part of this question asks you to give another way type 1 diabetes can be controlled. Now, remember, there's no real cure for type 1 diabetes because in type 1 diabetes, the pancreas does not produce enough insulin, or it might not produce insulin at all. So really, the treatment is pancreas transplant. However, whilst that would get you the mark, other marks are available because the thing with type 1 diabetes is you want to not just rely on injecting insulin, but you want your blood glucose levels to remain quite steady. So in order to help them remain steady, if you have a diet with less sugar, not just a diet, be specific and say a diet with less sugar, then your blood glucose concentration won't suddenly spike after a meal. It won't suddenly go up after a meal. If you do lots of regular exercise, then you will start making sure you're using up some of the glucose from your meal so that your blood glucose levels don't go too high. So all of these will give you the mark. Question 2. Again, which hormone controls glucose concentration? The answer is insulin. I will also accept glucagon because that helps control. It doesn't specify which one lowers blood glucose concentration and which one increases, so both are fine in this case. Next question, it talks about two people who have drank a 50-gram solution of glucose and they're measuring their blood every half an hour. So let's look at the graph. This is my caption. Now let's look at the axes of my graph. I've got time in hours and I've got the concentration of glucose in the blood here. Now you'll notice that A and B seem to have very different blood glucose concentrations, and whilst they both take the 50 grams of glucose at the same time, their bodies react very differently. This one seems to have a lot more glucose, this person A, in their blood compared to this one, and it also seems to last a longer time. So I'm already noticing differences before I read the question, which is, by how much did the blood glucose concentration in person B rise after drinking the glucose drink? If it says by how much, I need to compare person B before and after their drink. So before the drink they had 88 milligrams of glucose in their blood, and after the drink they had 99 milligrams. So the difference between 99 and 88 is 11 milligrams, so that's going to be my answer. Now remember, to help you work this out, you should always write down the number by the axes to remind you, and you should also work out what one box represents so that you don't make a mistake with labeling your axes. 2BII asks that I look for two pieces of evidence from the graph to support the doctor's suggestion that A has diabetes. So if we look at the graph, there's a couple of things that we notice. One, throughout the entire investigation, A has a higher amount of blood glucose than B. So that would give you the first mark. A has a higher blood glucose concentration at all times. Now that would get you the mark, or instead of that you could say pick any two pieces of data for the same time. So maybe you could pick at 1 hour what was their blood glucose concentration and say A's was higher at, and quote the figure. At any point would do. The second mark is for saying a larger increase in concentration after the drink. So if we look at person B, theirs rose by a little bit, but person A's rose by a lot. Again, you could either say it generally, or you can give the numbers to prove how much it increased by. So whilst we worked out that person B's rose by 11 milligrams, A's rose by 24 when you compare the two data points where they drank the 50 milliliters of glucose. There are other things as well you can say. There's a faster or a steeper rise for person A. We can see person B's glucose levels increased at kind of this gradient. However, with person A, it's a much steeper gradient, so it rose more quickly. And the fall is slower. So if you look at B's, their blood glucose was back below usual levels by 2.5 hours here, whereas for A it took a lot longer to fall back to their usual levels, probably between 3 and 4 hours. So it took longer to return to normal, or there was a slower fall. Again, comparative language is necessary in your answers. 2BII. Why is it that blood glucose concentration falls? Well, there's loads of reasons why your blood glucose levels go down. Either insulin was released, and that causes, remember, the glucose to go into the cells, or the glucose was used in respiration, or the glucose was converted to glycogen. Any one of those will do. Okay, number 3 is about reflex action. That is a nervous system response, like as you can see from the diagram. We'll learn more about this in year 11. The control of blood glucose or blood sugar level is an example of action controlled by hormones. Give 2 ways of reflex action shown here when someone's accidentally putting their finger onto a pin. How it is different from an action controlled by hormones. Well, if we think about it, when we know that when we put a finger on a pin suddenly, our hands will suddenly move away, and that is a reflex action. So how is this different to a hormone, for example, blood glucose levels? Well, reflex actions are quicker. So if reflex actions are quicker, again, I've used comparative language. We know this because, I mean, look at this graph. The time it takes hours for your blood glucose levels to be monitored and controlled by your pancreas. Hormones are slow acting. Nervous system is very quick acting, so it's quicker. And secondly, the effect lasts a shorter time. Once your hand's moved away, that's it. A few milliseconds it took for you to move your hand away. However, the effects of hormones last longer. It can last up to hours because the hormones continue to circulate in the blood and have their effect. Other answers that are accepted are here. So you can also say hormones involve blood and system, and the reflex action involve neurons or nerve cells. You can also say they involve impulses, whereas hormones involve chemicals. And you can say reflex affects one part of the body, whereas hormones affect maybe more than one part of the body, because it might affect all cells or a whole group of cells. Right, number 4. This question is about the nervous system. Describe the function of receptors. You just need to know that receptors detect stimuli and they convert information to an impulse, an electrical impulse that is carried by the nervous system. So it does 2 things. It detects the stimuli, so it might be a change in temperature, it might be some pressure, it might be pain. Those are detected by the cells in the skin, the receptors, and then that's converted into an electrical impulse, so the message can be sent forwards. Next, 4 easy marks for 2 effectors. Now, effectors carry out a response, a nervous response. So muscles, what's their response? They contract. Glands, what's their response? They secrete hormones. You could say release hormones instead. 4BII. Why is it important to control body temperature? It's important because it helps maintain temperatures at which enzymes work best. You could also say that it prevents enzymes from denaturing. So it prevents enzymes from denaturing. And you could also say that it prevents damage to cells as well. Okay, number 5. Now, this number 5 has a few bits which are for the higher paper only, so you may not have studied this necessarily, but if you have, then great, follow along. If not, you can pick up some new ideas that you may not have learnt yet, but may learn later on. So, the hormone released when the blood glucose concentration is too low is glucagon. Remember, the way to remember that, the little trick, is if the glucose is gone from the blood, in other words, if the blood sugar levels, the blood glucose levels are low, then glucagon is released. When is glucagon released? When the glucose is gone. If the glucose levels are too high in the blood, insulin is released. So that brings us nicely on to our next question. Explain how the 2 hormones keep the blood glucose concentration at the correct level. So you need to give the scientific reasons why. So the way you need to think about this is, well, first you need to talk about insulin, then you need to talk about glucagon and what they do and how, and then you can talk there's a 5th mark for kind of an overall picture. So, if blood glucose concentration is too high, insulin is released from the pancreas and causes glucose to move from the blood into the cells. That's the 1st mark I've got for saying what happens when blood levels, glucose levels are too high. And the result is blood glucose levels will fall. So I've already explained how we keep it at the correct level, the blood glucose concentration. I've talked about what happens when it goes too high. Now I'm going to talk about what happens when it goes too low. If your blood sugar levels fall, then glucagon is released by the pancreas and causes glycogen to break down in the liver and the muscle cells. And because glycogen, remember, is just lots of glucose molecules joined together, if they are broken down into lots of glucose molecules, they can be released into the blood, and the blood sugar levels will go back up again, because remember, glucagon is released when their levels are too low. So glucose enters the blood and raises levels again, and this is called negative feedback. Now this bit we may not have heard about yet, but we will study in more detail in year 11. Whenever something in homeostasis is about, if the levels get too high, then the body responds to bring the levels back down, or if the levels get too low, the body does something to bring the levels back up to a normal level, then we call this whole process negative feedback. So make sure you talk about negative feedback and use that phrase if you're ever asked a question about when the body responds to something in the body becoming too high or low by decreasing or increasing its levels. We call that negative feedback. So that's the 5th mark. Add that in if you did not get that. The final part of the question is about some recap from things we did last year about DNA. First, describe the structure of DNA. Well, we know it's a polymer. What is a polymer? A polymer is a long molecule made up of lots of repeating units. Okay, so if you didn't know that, it's a long molecule made of repeating units joined together by covalent bonds. So DNA is lots of these repeating units joined together, and we know it's made of 2 strands. We've got one strand and the second strand, like a ladder, and we know it's twisted in a double helix. Any 2 of these would have given you 2 marks. Finally, we asked to describe how DNA controls the structure of a protein. So remember, all DNA does is it is a code, so it contains a code. Let's say this section of the DNA is a gene for eye color. Then this gene will have the instructions as a code to make a sequence of amino acids. So maybe this is the sequence of amino acids it's coding for that will make a protein that is, let's say, blue in color, that will give a person blue eyes. Or that sequence of proteins will be brown in color and it will make the brown pigment that makes someone's eye color brown. So how does DNA control the structure of a protein? Well, DNA contains a code that is used to sequence, and that's an important word, sequence, to sequence amino acids together to form a specific protein. Okay, add up your marks out of 30 and put the total at the front.